Meiosis has one purpose, the production of gametes, your body's sex cells. Unlike mitosis, meiosis creates daughter cells with half as many chromosomes as the parent cell. Meiosis turns a diploid cell with two sets of chromosomes into two haploid cells with one set of chromosomes each. Before beginning meiosis 1, a cell will undergo interphase, in which the cell goes through three phases. In G1, the cell grows. In the S phase, the cell replicates its DNA, and in the G2 phase, the cell prepares for division. Next, the cell begins meiosis in prophase 1. In this phase, chromosomes begin to condense, and they pair up with their homolog partners so that they align. This process is helped along by the synaptohemal complex, a protein that holds homolog pairs together. The chromosomes can then cross over each other, allowing homologs to exchange parts of their DNA. This can be seen in the chiasmata, cross-shaped structures where homologs are held together. Next, the cell begins metaphase 1. Homolog pairs attach to microtubules from opposite poles and line up at random orientations along the metaphase plate for separation. Next, in anaphase 1, homologs are pulled apart. Sister chromatids then move to the poles at opposite sides of the cell. In telophase 1, the chromosomes are on opposite sides of the cell. At this point, cytokinesis can happen, separating the cell cytoplasm and creating two daughter cells. The cell can now move into meiosis 2, the next step in creating an egg or sperm cell. The cell begins this phase as the cell that just completed meiosis 1, a haploid cell with one chromosome from each homolog pair, made from two sister chromatids. Meiosis 2 will separate these sister chromatids, creating 4 haploid cells. Next, in prophase 2, the centrosomes move apart and the spindle is formed between them. Next, the spindle microtubules start to attract the chromosomes. In metaphase 2, the chromosomes once again line up along the metaphase plate and attach to the microtubules. In anaphase 2, the chromosomes are pulled apart into the individual sister chromatids and moved towards the opposite poles of the cell. In telophase 2, the nuclear envelope returns, the chromosomes recondense, and cytokinesis can once again happen. Once the process of meiosis 2 is complete, 4 haploid cells with unique DNA now exist. In males, all 4 of these cells turn into 4 individual sperm cells. In females, a larger amount of the cytoplasm and organelles end up in one of the 4 cells. That one cell will be the egg, and the other 3 cells become polar bodies, which are useless in humans. To create life, one haploid sperm cell will fertilize a haploid egg cell, creating a full set of chromosomes for the offspring.